Good evening, everyone. Myself Vishal. Today we are going to discuss regarding the grams staining that is prescribed in the B Pharmacy third semester syllabus in the pharmaceutical microbiology. So after completion of this presentation, we or else the student will able to learn this kind of the objectives, that is defining the gram staining, purpose of the gram staining, and after that the student will able to understand regarding the staining techniques regarding the gram staining. That means like a student will able to understand the significance, purpose, and what are all the stains involved in the gram staining. Okay. So before starting this staining techniques, we shall able to understand what is the purpose of the gram staining. Okay. So basically, in 19th century, in the Grams Christian, the scientist has designed this gram staining in order to differentiate between the gram positive and gram negative bacteria, because in the pharmaceutical industries there are a lot of antibiotics and to be differentiated, because each kind of a bacteria has a different what I can say drug resistance. Okay, so we need to check which kind of the bacteria has the which drug resistance or not. Due to that thing, due to this thing, we are able to differentiate the morphology, anatomical features, and the cell wall and the different things involved in the bacteria. So now let us understand the first procedure, then I'll explain the principle of the gram staining. Okay. So in the first step, students, we need to take an unknown sample. The unknown sample, that means like previously nutrient broth has been prepared. For that, we can able to add any microbial sample, and we need to incubate in the biological oxygen demand for the 24 hours. After incubation of a 24 hours, we need to take the nutrient broth and we need to perform the experiment. First step, we need to take the glass slide. On that glass slide, we need to take the broth containing the microbial sample. So before that, we need to turn on the burner, two burners, and we have to start making the thin smear. What do you understand by the meaning of the thin smear? Thin smear, in a sense, like we have to take an inoculum loop and we have to dip it into the broth. After that, we are going to make a thin smear. After performing the thin smear, we need to do the fixation of the smear. How we are going to do the fixation of the smear? By passing on to the Bunsen burner. That means, students, you can see the slide, the glass slide which you have taken. On that, we need to pass it to the Bunsen burner for a period of 15 to 30 seconds. After that, there will be the formation of a thin film. That thin film is only called as the smear. In the first step, students, you can see in the diagram, you can see like addition of the crystal violet. What is this crystal violet? This crystal violet, you can see this crystal violet which is there, it is called as your primary stain. Okay, what does this primary stain will do? If you take any kind of the bacteria, it will give rise to the purple color. Okay, after addition of the crystal violet, that is a primary stain, let it allow for the period of 30 seconds. After allowing for the period of 30 seconds, then take a tap of water in the beaker, then wash it, and after 30 seconds we need to add the second thing. What is that second thing? That is the mordant. In the gram staining, the purpose of adding the mordant is for the fixation of the primary stain. Now if I am taking the name mordant means it is the grams iodine. So what is the composition of the grams iodine? The composition of the grams iodine is potassium iodide and iodine. So this mixture composes of the iodinic solution with the potassium iodine. The main advantage of adding the grams iodine, that is the moderant, is whatever the primary stain you have added in the first step, that is a crystal violet, all the crystal violet, whatever it is there on the prepared on the sample, it will attach. So that is the purpose of adding the moderant. So usually we'll be adding the 1 or 2 drops, whether it is a primary stain, whether it is a moderant, we'll be adding 1 or 2 drops. After adding 1 or 2 drops, then what we need to do, we have to again allow to add for the 30 seconds. After waiting for the 30 seconds, again we need to take the water and we need to wash. After washing, then the next thing is the main step, and is we are going to add the alcohol. Alcohol, that is 95% in the gram staining, is used as a decolorizing agent. So here in this step we are going to add the decolorizing agent. Okay, so this will decide whether it is a gram positive or the gram negative species. Followed by this, again we need to wait for 30 seconds. We need to wash. After washing, we need to add the last stain, that is the counter stain. Counter stain means that is the saffronin. So after adding the 1 drop of saffronin, we need to again wait it for the 30 to 40 seconds and wash it with the water. Then we need to examine under the microscope. Okay, so this is the procedure. Okay, so now exactly morphological and anatomically, let us try to understand the what is gram positive and gram negative bacteria. Suppose your sample may be gram negative. So in the microscope you can see it as a pink color. If it is a gram positive, then you can see that as a purple or as the violet color. Okay, so students, let us try to in this slide we can able to see. So the gram positive which is there, that is in the purple color, and the gram negative which is there, that is in the pink color. So students, you can see this is the image of a microscopic, where you can see the purple dots which is representing, that is the gram positive bacteria, and the pinkish which is there in little bit like as a rod shape, that is called as a gram negative bacteria. And you can see students, if I can show you the cell wall, so this will be the cell wall of the gram positive and gram negative bacteria. So why gram positive will retain the purple color and gram negative will retain the pink color is due to the cell wall. That means in the gram positive bacteria and the gram negative bacteria, main thing it is a cell wall. So this cell wall, that is made up of the peptidoglycogen layer, will decide the color of the bacteria. Okay, now here we can able to see like what exactly happens. So students, in this slide we can able to see that is a gram positive cell wall. We can see, if I am going to take the structure, the first thing we can see, that is the teichoic acid and the lipoteichoic acid. The purpose of teichoic and the lipoteichoic acid in the cell wall is to give the rigidity. That means, already I said, in the gram positive bacteria, it is in the purple color. First, what you did, students, you added the primary stain. Next, you added the gram iodine. Next, what you did, you added the decolorizing agent. Then next, you added the saffronin. Here, what happens? Here in the peptidoglycogen, see students, this is the very what I can tell, thick peptidoglycan. This peptidoglycogen layer in the gram positive cell wall, it is very rigid, that is strong. Here what happens, when you add the what I can tell, primary stain and the gram iodine, it will form crystal violet and iodine complex, such a way that due to its rigid and very thick peptidoglycogen layer, it doesn't allow the ethyl alcohol to pass. In the next step, what we'll be adding, students, we'll be adding the alcohol, right? Now what happens due to its rigid pores, you can see in this diagram, whatever the alcohol you add, it will not allow. In the next step, what you are going to do, you are going to add the safranin. No, it doesn't allow. Why? Periplasmic space is also present, and plasma membrane integral protease is present, and teichoic acid, what it will do, students, it will is very rigid. It supports the peptidoglycogen layer such a way that it doesn't have any pore space to pass. Such a way that after you are adding the gram sardine, after you are going to add this decolorizing agent, what happens? Due to the very rigid peptidoglycogen layer, there is no pores to pass on, so nothing it will enter. Coming to the gram negative bacteria, you can see, students, it is lipopolysaccharides is there, that is the outermost layer. This is absent in the gram positive cell wall, and we are going to have the outer membrane which is also absent in the gram positive cell wall. Periplasmic space is there, and you can see, students, very thin peptidoglycogen layer is there. See, peptidoglycogen is a very important thing that can able to define the cell wall. Since the peptidoglycogen is very thin and outer membrane is there, what happens in the third step, when you add this alcohol, the alcohol washes the crystal violet and the iodine complex and it will become colorless. Why it doesn't happen in the gram positive cell wall? Because it had rigid peptidoglycogen layer and it has rigid pores. But what happens in the gram negative cell wall? Peptidoglycogen is very thin. It is having the outer membrane. It is not having any support of the teichoic acid. What happened? Decolorizing agent has entered into the peptidoglycogen layer. It has washed the crystal violet and iodine complex, and what happened? Gram negative has become the colorless. In the last step, what you are going to do, students, you are going to add the counter stain, that is safranin. Since the crystal violet and the iodine's complex has been washed off by the alcohol, in the last step, what you are going to add? You are going to add the safranin. That safranin will convert the gram negative bacteria into the pink color. Okay, so this is how, students, the gram positive and gram negative cell wall are going to get the color. The main thing is cell wall. In that cell wall, what it is important? Peptidoglycogen. One it is having the rigid peptidoglycogen layer and one it is having the thin peptidoglycogen layer. See, students, and already I said that is in the gram positive and the gram negative. If I am going to see the chemical entities, lipoprotein is absent in the gram positive and it is present in the gram negative. Lipopolysaccharide is also absent in the gram positive. It is absent in the gram negative. Peptidoglycogen is there. Both also gram positive is there, gram negative is there. Rigid peptidoglycogen layer in gram positive and very thin peptidoglycogen layer in the gram positive. Polysaccharides is there in gram positive it is not there. Proteins might be present in the gram positive or gram negative. In the gram negative it is not there. Clear? And as you can see, students, in the gram negative bacteria, you are going to have the uniform inner cell wall, that is 2 to 3 meter wide, and outer layer it is 8 to 10 meter wide. In the gram positive, students, there will be the inner and rigid cell wall where it is having the teichoic acid. Already I have explained in the table, and you can see, students, the example of the gram positive and gram negative bacteria. In the gram positive it is Staphylococcus aureus, Pseudomonas aeruginosa, Streptococcus genus, and in the gram negative Neisseria gonorrhoeae, Stipulium voluntas, and the Neisseria menendez and E. coli. Here also you can see this classification, and these are the references. Hope so, students, you understand the topic. Thank you.